think we can start.
Let's start, okay.
I just thanks to all of you to be here and to those that are remote. I will leave the floor to Fabrizio Capaccioni to introducing our speaker of today, Jonathan Lunine. I would just remember to the people that are here and remotely that at the end of the talk there will be time for questions. And for those that are remote, maybe it would be preferred to ask to raise the hand so we can follow the queue to eventually question from remote. So Fabrizio, I give you the honor.
What I can say, I'm with great pleasure and I'm honored to have Professor Lunine Jonathan here today. Paraphrasing Allen Ginsberg, I would say in a positive terms, in a positive sense, one of the best minds of our generation in planetary sciences, and not only planetary sciences, astrobiology, astrochemistry. So there are many activities in which Jonathan is involved, has been involved, and will be involved, because maybe not all of you know that he has been recently not nominated, how should I say, appointed as chief scientist of JPL. So he will take care of all the activities, scientific activities in JPL. And he has been the recipient of many awards, the Urey Prize of PBS, the Zeldovich Medal of COSPAR, and the Cassini Medal of GU. So he has a long story and a beautiful future. And we were here to listen to his ideas on life in the outer solar system. Right. Thank you very much.
Thank you, Fabrizio. Well, thank you, everyone. It's really very emotional for me to be back here, because over the period from 1998 to 2011, I spent 2 sabbaticals, several summers, and then a period of 2 years when I was teaching at Tor Vergata, spending much of my time here at the Institute. So it's very much like coming home. And in that regard, I want to dedicate this talk to my good friend and our colleague, Angeletta Corradini. And that is one of her famous phrases that came from Fabrizio's memorial of Angeletta that you wrote just shortly after she passed away. I remembered Angeletta now, especially not only because I'm in Rome, but when Angeletta passed away, she was 65 years old. And my 65th birthday was yesterday. So I became 65 yesterday. And so, you know, same age, and then you think about that quite a lot. For those of you who are perhaps more early career, Angeletta was a brilliant scientist, an extraordinary leader who really was the most important in developing planetary science and planetary space instrumentation here in Italy. And she was an amazing reader of human character. So she was the person who was able to explain to me, and of course she was never at all timid about explaining, what my fundamental flaw is. I'm not going to tell you what it is, but we had a discussion one morning when it was relevant, and she just said, this is it. And she explained it, and it was right on. So Angeletta was a remarkable human being and passed away much too young. So this is a field that she would have been, and in fact was very much interested in and a part of. So I'm looking at my cell phone because that's my clock. Okay, so the subject of the talk today is looking for life beyond the Earth. And there are really 3 ways to do this, and they're listed here. So one is to look for advanced, intelligent, self-aware, technologically capable life elsewhere in the galaxy. And the term for that typically has been the search for extraterrestrial intelligence, mostly done by radio. It is very much a roll of the dice, because if you find a communication, then Earth history has changed. If you don't find a communication or a signal, you don't know why that is. And there are some possible reasons why that people like to speculate about. But fair to say, over the period from the late 1950s, when this was first proposed, actually Cornell, a quantitative radio search, up to today, there really has been nothing. Then there's the search for life by looking at planets around other stars with telescopes, with spectroscopy, and that's a quantitative scientific experiment. You can look for gases in atmospheres that are evidently out of, not only out of chemical equilibrium, but in such a way that you would expect one of the gases to go away very quickly. So on the Earth, we have both methane and molecular oxygen in the atmosphere. Those are very unstable. Of course, Mars does too. That was an interesting discovery that was first actually made on Mars Express, really, and then later by the Curiosity rover. The problem with that approach to searching for life is that it's very difficult to demonstrate that one has actually found life by looking for a molecule in an atmosphere. There are certainly molecules that likely are not made in nature. One recent example is in the paper by Medusadon and colleagues, where they think they've discovered dimethyl sulfide in the atmosphere of a sub-Neptune planet. But then it becomes the challenge of the chemist and the geologist to show how you might be able to get that in a non-biological way. And so there really is no way to study cells or the biochemistry directly, even though this is an important way to do things to search for life. What I'm going to talk about is what I would call the direct search for life, and that is to search for life in our solar system by going to environments which evidently can support life, that are habitable environments. And we expect that this life would be microbial. Microbial life began very early on our own planet. It took many billions of years for multicellular animal and plant life to appear, but simple microbes appeared perhaps within the first few 100 million years of the evolution of the Earth. So we expect microbial life perhaps has arisen anywhere that has had a habitable environment for a few 100 million years, maybe even less. With the search for microbial life, you can do direct chemical tests. You may not even have to drill into the crust or the ocean, as I'll explain for the case of Enceladus. And the potential for direct confirmation is very high. It's possible that one might actually acquire even a cell from cellular life, but at least the chemicals that are being made from life and that are present in patterns that would tell us that the origin of those chemicals, that chemical system, is biological. So that's, I think, the most promising way to do this. And then the question is, how common might life be if we have a habitable environment somewhere else, like the ocean of Europa or the ocean of Enceladus? What can we say that very definitely life would arise? And at this point in time, the answer is no, no one can do that because there's no actual model for life, how life formed on the Earth. There's no set of steps that one can actually list in the way that one can list the steps for the formation of a planet, for example. And so one speculation is illustrated in this figure that the origin of life might be very difficult. That life, first of all, requires an environment, if we look from the top going downward, that requires some processes that decrease the chemical diversity of the system, that select out of the multitude of possible chemical compounds that could be made from carbon, for example, the ones that in particular are going to produce biological molecules. Molecules are useful for biology. So for example, our DNA and our RNA, deoxyribonucleic acid and ribonucleic acid, the backbone of both of these is a sugar, deoxyribose for DNA and ribose for RNA. But in an abiotic chemical environment, either even in interstellar space or on a planet, sugars of different varieties are made with equal propensity. And so selecting out of this mass of different kinds of sugars, deoxyribose or ribose, most likely ribose, it's not clear how that happens. It's not clear what that process is other than biology itself. So there's some kind of a bottleneck there, and that bottleneck may perhaps be a very difficult one. We don't know. And so being able to look in environments elsewhere in our solar system that could support life and see if there really is life there will tell us how severe that bottleneck really is. And that in turn could tell us whether life is very common elsewhere in the galaxy. So there are many places to potentially look for life. One of the most exciting is Mars. Yeah, most of the slides are not too dark, so this should be okay. One of the most interesting is Mars, because Mars had an Earth-like environment in the first half billion to billion years of its history. We know that from Mars exploration. But I'm not going to talk about that, because there we are almost certainly looking for, is that better or worse? I don't know. Now you see me in silhouette, so honestly, I think the slides will probably, most of the slides are brighter than that. Okay, but anyway, Mars is a place where we would look for evidence of past life in the environment. And Jezero Crater, for example, is a very good one. But likely, if life exists today, it's so deep below the surface that it would be hard to get a hold of. So I'm not going to talk about Mars. I'm going to talk about environments in the outer solar system where liquid water or other liquids exist, either on the surface or underneath the surface. And places where there are oceans of liquid water are places called ocean worlds. And the ones that we know about for certain are Ganymede, Europa, Enceladus, and Titan. And Titan also has liquids on the surface, liquid methane, liquid ethane. Callisto might have an ocean, but the evidence for that from Galileo is fairly faint. The JUICE mission will settle that question very definitively, but for now, Callisto is a question mark. In the case of Ganymede, that ocean is so deep below the surface that it will be difficult to access to look for life. And so I'm going to focus on Europa and Enceladus, or the liquid water oceans. Titan's ocean is also very deep below the surface, but interesting for Titan is the possibility of some kind of biology in the methane lakes and seas that are present on the surface. Let's see how I'm doing. Unfortunately, there's no way to stop the thing from going to sleep. Okay. So let's start with Europa. Europa is a moon of Jupiter. It's about the size of the Earth's moon. You can see in this figure Europa and our moon shown precisely to scale relative to the Earth. Europa is the second of the 4 Galilean moons, the Medicean stars, as Galileo Galilei called them, that are in orbit around Jupiter. And Europa is special because it's actually a moon that's mostly made of rock, as is the case for Io, the next one in. We know that from measuring the mass and knowing the size of these objects. The masses were measured by the flybys of Voyager. So, but Europa is not only rock. Europa has an outer layer of water. And the Galileo spacecraft, which orbited in the 1990s, was able to demonstrate that much of that water is actually liquid water and not water ice. So the outer surface, which is very bright, is water ice with small amounts of other compounds. But below that ice covering, as you see in that little diagram on the left, is an ocean the size of which is constrained because, again, we know the density of Europa. It's 3 grams per cubic centimeter. So that constrains the amount of water relative to rock and metal. We know that it must be mostly rock. And because also we have some information on the moment of inertia, and the fact that ice is less dense than rock, we would expect all of the water to be on the outside as a mantle around the rock. And so that dictates the thickness of that mantle. And then the estimates for the thickness of the outer part of that, the ice crust, gives us an ocean volume that is somewhere between 1 and 2 times the volume of all of the ocean water on the Earth. So the largest ocean in the solar system almost certainly is that of Europa. And there's very interesting geology on the surface, fractures that and ridges that crisscross each other. I'll show you an example of a feature that might be the result of crustal melting in a moment. So how do we know from Galileo that there is a liquid water ocean? Well, this actually comes from magnetometry, not from imaging or gravity. So this is an animation of Jupiter's magnetic field, and the field lines you can see emanating from Jupiter, and you see Jupiter rotating. The axis of Jupiter's magnetic field is tilted significantly relative to its rotation axis, and the Galilean moons orbit in a plane that is exactly perpendicular to the rotation axis, 90 degrees from that. As a consequence, the magnetic field is tilted relative to the orbit plane of the Galilean moons. And so what that means, and along with that, I should say that the field of Jupiter is not entirely dipolar, it has higher order components. So what all that means is that a body that is orbiting in the equatorial plane of Jupiter experiences a magnetic field that is varying in time with intensity. And if you look carefully, you can see that the field lines really are tilted and they're bobbing up and down. So this time variable magnetic field sweeps through Europa, and as a consequence of that, if there's any conducting material inside of Europa, that time varying magnetic field will generate eddy currents, electric field, and that electric field, the eddy currents, will generate an induced magnetic field. And it was that induced magnetic field that was detected by the Galileo orbiter as it made multiple flybys of Europa. And one could actually fit the geometry of that induced field and the strength of that field to allow one to conclude that the source of that field, the conductor, is not deep near the center of Europa. It has to be relatively close to the surface and fairly large in volume. And so the only candidate for that is liquid water, in fact, salty liquid water. So this is the way in which the presence of an ocean is inferred. And then the geology of the surface of Europa, which shows very few craters, there's one large crater, suggests that the surface, the crust, is being renewed all the time and is relatively thin, no more than 40 kilometers in thickness, overlying that ocean. So the crust also melts. And this is a feature that is in a part of Europa that's called chaos terrain. It's an image that was taken not by Voyager or by Galileo, but by the Juno spacecraft, which made a very close flyby of Europa 2 years ago. And this is an image that was actually taken in Jupiter shine. The sun was over on the other side, so this part of Europa was in shadow, but it was facing the lit part of Jupiter. And the camera that took this image is actually a low light navigation camera that's designed to take images of stars against the night side of Jupiter so that the orientation and position of the spacecraft can be determined. But it's a great camera for geology. And so you see this feature in the lower right. It's about 65 kilometers top to bottom. Heidi Becker is the principal investigator for the science portion of the Stellar Reference Unit for the science images. And we formed a little team to analyze this. We call this feature the platypus. And so I would challenge you to imagine the You see the two dark eyes there, and below the eyes are the bill of the platypus, and the body of the platypus is above that to the north. So there we go. So that feature is interesting because it was imaged not only by Juno, but also by Galileo from a different angle. So there's topographic data. And the body of the platypus, the part above, is actually slightly dome-shaped. And that's an indicator that this is a feature that probably formed when warm ice welled up through the crust, melted a region with salt, and that melted region then refroze, and as it refroze, it domed up. This is a model that was developed by Brittany Schmidt and colleagues about 12, 13 years ago. And then evidently some material that's not water ice was extruded onto the surface. So the two dark eyes are not shadows. There's some material that has been darkened on the surface. You'll notice, by the way, all these white spots on the image. And that's an indication of the very intense radiation field of Jupiter. So Jupiter's strong magnetic field, 20,000 times stronger than that of the Earth at the respective surfaces of these two bodies. That magnetic field picks up ions from the solar wind. Those then collide with surfaces to pick up more ions and electrons, and so the radiation environment, the particle radiation environment, is very intense. And so not only does it produce white spots on the detector, but it darkens any material that's not ice, either a salt or organics, that are extruded onto the surface. And if you look at the upper part of the image in that double ridge region, you can see more of these dark spots. And those dark spots are very similar to what was found in Galileo orbiter imagery on another part of Europa and analyzed by Lunine and Matt Kedman. And you can see the similarity between these, and their interpretation is that this is material that is not water ice and has been extruded onto the surface and has been darkened by radiation, which means either organic material or certain salts. And so that is indirect evidence that ocean material comes out onto the surface and contains compounds other than water ice itself. And to strengthen that conclusion, one can look at data from Hubble Space Telescope and the James Webb Space Telescope, which have been used to map the surface of Europa. And both sodium chloride salt and carbon dioxide ice have been seen on the surface of Europa. Sodium chloride by Hubble and carbon dioxide by JWST, which are correlated with the geographic positions of these so-called chaos terrains. And so the fact that they're correlated and the fact that these chaos terrains are the most heavily fractured parts of Europa and probably have the thinnest crust is very strong evidence that ocean material is coming out onto the surface and then being radiation darkened. So that's a very positive thing for being able to explore the nature of that ocean underneath the crust. It's possible the material is coming out in the form of plumes or jets. And so, but even if it's not, being able to analyze these deposits with good near-infrared spectrometers will allow us to determine whether this material is, for example, organic molecules, what kind of organic molecules, what kind of salts besides sodium chloride, and therefore to determine whether the ocean is habitable, whether it has the nutrients necessary to support life. So the Europa Clipper is designed to do just that. It's been delivered to Cape Canaveral for launch in October of this year. It will not orbit Europa because of the radiation. It will orbit Jupiter and make about 48 flybys of Europa. And the instrument suite that's present, and these are just the acronyms for the instruments, range from gravity experiment, which I'm involved in, to magnetometry, which will more precisely determine the salinity of the ocean, moving upward from ice mag and grav to reason and EFEMIS, a radar and long wavelength infrared spectrometer to determine the structure and thickness of the ice and look for these warm pockets of ice that may be rising to the surface, and then remote sensing of the surface by the camera, ICE, the ultraviolet instrument, MISE, which is the near-infrared instrument, which I'm involved in, and then 2 mass spectrometers as well. So this will give us, what we hope will be the identity of some of this material that is coming out from the interior, deposited on the surface. Tell us whether the ocean really does have the material necessary for habitability and whether it's worth going back with a lander. At the same time, the Jupiter Icy Moons Explorer, which is already on its way to the Jupiter system, the ESA mission, is similarly instrumented. We'll make 2 flybys of Europa, many flybys of Callisto, and will eventually go into orbit around Ganymede. And Ganymede, based on data from the GIRAM instrument, which was the Aussie instrument, is the Aussie instrument on Juno, in a paper by Tozy and colleagues, there's very strong evidence for non-water ice material on Ganymede as well. So that ocean may as well contain the material necessary for habitability. So the Jovian system is very exciting, but we can't say yet today how habitable those oceans are, but hopefully 10 years from now, it will be possible to say yes or no, whether these are places which may have life in those oceans. Yes, there are the instruments on JUICE, and there's also something called PRIDE, which is a VLBI experiment that will improve the ephemerides of all of the Galilean moons as well. Okay, let me move on to Enceladus. So Saturn's moon Enceladus is nowhere near as large as Europa. There it is shown actual size, but it is, as it turns out, a very active moon. It has a plume of gas and dust and ice pouring out of the south polar region, probably the result of tidal heating either now or in the very, very recent past. It's not quite in an orbital resonance with Dione, the way that Europa is in resonance with Io and Ganymede, but it's close to that resonance, and so it may go in and out of that resonance. So it has an ocean too. And that ocean was discovered by Cassini and was directly explored thanks to the presence of the plume. So here's some beautiful pictures taken by the Cassini mission, which was the joint NASA, ESA, Aussie mission that arrived at Saturn in 2004 and ended in 2017 with the depletion of its propellant, and it was then burned up in the atmosphere of Saturn to prevent it from hitting Enceladus or Titan. You can see in the center, upper center, one of the early pictures of the plume, backlit sunlight revealing that plume. In the lower left, you see the plume is made of a series of jets, or possibly a curtain of jets coming out of fractures. The large-scale fractures are shown in the upper left. The finer detail at the 100-meter scale is shown in the lower right. You can see the surface is very convoluted at the South Pole. And then in the upper right, you see the E-ring, which is made of material that is coming out of Enceladus because it's a small moon. The gravity is very low, and so particles up to several microns in size actually get injected into orbit around Saturn to form this ring. That's important because much of the analysis of the chemistry in the plume came not only from the plume material itself, but from sampling the E-ring as well. And Cassini carried 2 mass spectrometers. Neither was designed for Enceladus because no one knew there was a plume of Enceladus at the time, but they were designed to look at dust in the Saturn system and compounds in Titan's atmosphere. But they turned out to be very useful because it was possible after Cassini discovered this plume and after some study of the safety of flying through the plume to actually fly Cassini through this plume at an altitude as low as 49 kilometers and sampling that material directly, which means that Cassini was sampling the material from the inside of this moon without having to land or drill or do anything like that. All right, so mass spectrometers, just as a brief review, separate species by mass. There are many different kinds of mass spectrometers. This is a quadrupole mass spectrometer where you have 4 metal tubes to which a time variable electric field is applied and is phased differently on the different tubes so that you can control when particular ions of a given mass reach the detector. That's one kind of mass spectrometer that flew on Cassini. That was the INMS. The other kind is called a time of flight mass spectrometer, in which, and I apologize, you can't see me on the Zoom, but you have 2 plates, 2 charged plates inside the instrument. The ions, and this is always done by ionizing the species that you want to analyze. The ions move in between the plates, and the time it takes to move from one plate to another and back determines the mass of the species. Technically, the mass over charge, but the ionization is almost always singly ionized, and so that then by default gives you the mass. So what did the mass spectrometers do? Well, first of all, they were part of the set of data that convinced everyone that underneath the icy surface of Enceladus, and I should say that Enceladus has a density of about 1.6 grams per cubic centimeter, so it's about 60% rock and 40% ice, but some of that ice is liquid water. So the mass spectrometers were part of the evidence that some of this water is really liquid under the surface, that there's an ocean. And so the mass spectrometry part of that evidence is shown here in a paper by Frank Postberg and colleagues. He's now at the Free University of Berlin. And it is a mass spectrum from the dust mass spectrometer, the cosmic dust analyzer, the time of flight mass spectrometer. And so what you see here is mass in atomic units on the x-axis, the amplitude of the signal on the y-axis, and the identification of the species based on their mass. You see sodium, potassium, chlorine as well, some carbonate, as well as OH. And so this is a salty ocean. Now, Cassini could not do the magnetometry detection at Enceladus that Galileo did at Europa, because Saturn's magnetic field is very much a dipole and is very much coplanar with the rotation axis. So there's no variation in the magnetic field that a conducting body feels in orbit around Saturn. So this detection is completely different. So salty water coming out of Enceladus, flash freezing to make ice particles up to several microns. Another form of evidence that was very compelling for a global ocean came from the libration, the observed libration of Enceladus. Enceladus is in an elliptical orbit around Saturn. It is synchronous, but because the orbital velocity in an elliptical orbit changes from the periapse to the apoapse, a synchronous orbit is actually not one in which the same face points toward the central body all the time. In fact, it points to the empty focus. And so, as seen from Saturn, Enceladus librates back and forth, just as the Earth's moon does, as seen from the Earth. Part of that libration is just the geometry of the orbit and the orbital speed, and part of it is a forced libration due to the fact that Enceladus is not entirely spherical, and so there's a torque that gets applied which increases the amplitude of the libration. That libration for Enceladus, as measured by Cassini, turns out to be 3 times what you would calculate for a body of the mass of Enceladus in its eccentric orbit around a body with the mass of Saturn. So the only way to get that kind of larger amplitude is to imagine that the upper part of Enceladus is slipping back and forth relative to the bulk of the interior. That is, it's not completely connected to the interior, that there's a frictionless, not completely frictionless, but low friction layer in between the upper icy crust of Enceladus and the rocky material at the base of what we now know to be liquid water. So that model then is the only one that really explains this large libration amplitude for Enceladus, that there is a liquid in between the ice crust at the top and the silicate portion further down, deeper down. Because what's coming out in the plume is water ice that has salt in it at the level of 1 or 2%, argues that the liquid is liquid water. So you put those pieces together along with other evidence, and you get a liquid water ocean as the source of that plume material. So what else do the mass spectrometers tell us? They tell us that there are organics in the Enceladus ocean. This is another spectrum, and I'm sorry, the y-axis label should be amplitude. Time of flight in microseconds corresponds to the atomic mass, which is given for each of those peaks. And if you do a little bit of simple arithmetic on the little peaks on the right-hand end of the graph, you find that they are separated by 12 or 13 mass units, which is a carbon or a carbon-hydrogen. And so that whole region is, those peaks are due to organic compounds. The mass resolution of this cosmic dust analyzer is not good enough to tell you what kind of organic compounds. So the label there stands only for high mass organic compounds, but not what they are. That requires a much better, higher mass resolution mass spectrometer than what Cassini could carry. The cosmic dust analyzer also found in the E-ring silicon dioxide. You see in this figure the amplitude again versus time of flight. You see the peak of silicon, Si, and the peak of oxygen, O. And you can see with your eye that it is in a 1:2 ratio, so SiO2. This is not beach sand because these are very tiny nanograins, nanometer-sized particles. And this mass spectrometer, because the dust actually collides with the collision plate in the instrument, can measure the mass of the particles. And these are remarkably narrow in their size or mass range, as if they are some sort of a colloidal suspension in the ocean itself. And so Sean Shu, who was at LASP at the time, who led this paper, postulated that this was evidence for liquid water interacting at the base of the ocean with rock to leach out, to first react and form SiO2, as in this equation down below, and then to leach that SiO2 out into the ocean. And as the ocean, as that water moved from the hotter rock to the colder near surface, that SiO2 would come out of solution into a colloidal suspension and be shot out of Enceladus along with the rest of the plume. What that implies, if this reaction is correct, is that there should also be hydrogen, H2, coming out in the plume, because that's another product of the reaction of water with the silicate to make magnetite, aqueous silica, and hydrogen. And lo and behold, the other mass spectrometer, the INMS, on its closest fly through the plume, detected molecular hydrogen and was able, this is a map of where it was detected, was able to determine that it's correlated with the fractures. So all of this hangs together. There's a liquid water ocean with salts, organic molecules, and chemical reactions between the water and warm rock at the base of the ocean. And that is very similar to what one finds in what are called off-axis hydrothermal systems on the Earth, where in fact various forms of life exist and feed off of the chemical energy that's provided there. So the Enceladus ocean has the basic ingredients for life. Now, very recently, Cassini data were reanalyzed by Postberg and colleagues. One of the questions was whether phosphorus would be present. phosphorus is critical to life. It's critical to the energy exchange in our cells through ATP and ADP. It's critical for the bonding of DNA and RNA. And it's very interesting because it's one of the key molecules of life, but it's the one that has the lowest cosmic abundance of those key elements, the so-called CHNOPs, or CHNOPs. When you ratio the abundance in life to the cosmic chemical abundance, phosphorus comes out highest. So it's utilized by life, but it's not quite so abundant on a cosmic chemical basis. And in a paper published just last year, Frank Postberg and colleagues doing further analysis of the time of flight data, the CDA data, found phosphates in the ocean of Enceladus. So if you were to throw microbes into this ocean, they would survive. We don't want to do that. So confirming that was a calculation that was done by Chris Lunine at Southwest Research. What he did was to take the data from the two mass spectrometers, which provide us with the abundances of hydrogen, methane, carbon dioxide, and water. They also provide a pH and acidity for the ocean through the chlorine compounds that you saw in that spectrum, and abundances, as you see in the upper left, from the gas mass spectrometer, the quadrupole mass spectrometer, of carbon dioxide, methane, ammonia, and so forth. And he took the hypothesis that the methane and the carbon dioxide balance is determined by the equation you see in the upper right, which is in biology called methanogenesis. And he asked the question, are the abundances of these species consistent with the presence of microorganisms? You could make CO2 and CH4, you could exchange them non-biologically. But if you calculate how far from equilibrium this system is based on those abundances, and that's the apparent affinity, which is the y-axis here, where 0 is chemical equilibrium and a positive number means energy is available. Biological systems on Earth exist in chemical systems that are out of equilibrium. The affinity is not 0, but it's never much larger than 0, because if there's food to eat, microorganisms will eat that and tend to bring the system back down to equilibrium. So he did that calculation, and the blue box represents the allowed region for Enceladus based on the abundances of CO2 and CH4, which give you the result on the y-axis, for the given amount of hydrogen relative to water, which gives you the value range on the x-axis, and then constrained by the pH allowed by Cassini. And the region that Earth microbes exist in is given by the double-headed arrow labeled Earth microbes. So the value is a little bit high. Most terrestrial ocean systems, microbial systems, drive their environment closer to equilibrium, closer to 0 than what is shown here, but there's still an overlap. And so the Enceladus Ocean could possibly indeed be a home for microbial life. So how do we determine whether that's the case? Well, it depends very much on what you want to learn. So we've learned already at the bottom of this pyramid that the ocean of Enceladus is habitable. If we want to understand what kind of life is there, if there is life there, we want to analyze cellular structures. Those are at the top of this pyramid. That's the most difficult thing one can do. But in between are tests of the presence of life that can be done with relatively simple instruments. And so the next step for Enceladus is not to go and try to collect cells and determine what the cellular structure is, because that would be very difficult to do, but rather to go back, fly through the plume again, but use better present-day technology mass spectrometers to look for the presence of biological molecules that can't be detected with the Cassini mass spectrometer. So what's wrong with the Cassini mass spectrometers? Well, nothing, except that their mass resolution is quite low. So in the left-hand chart, you see the signal for the gas mass spectrometer versus mass. And the mass resolution was such that you could tell whether a molecule had, for example, mass 28 or mass 29, but that leads to a lot of ambiguity. So for example, carbon monoxide and nitrogen and ethylene, C2H4, all have mass 28. And so the mass 28 signal could be any of those. Laboratory mass spectrometers allow you to determine the fractional mass, and the fractional mass is diagnostic because when atoms combine together to make molecules, the binding energy in their nuclei gives you either a greater or smaller, usually a smaller mass, than the individual atoms. So a molecule of N2 or a molecule of CO does not have the unitary mass 28. It has a mass deficit or excess, which represents the nuclear binding energy. And mass spectrometers today can detect that. The mass spectrometer on the Europa Clipper mass specs has the resolution to do that. And that's what my group working at JPL would like to fly to Enceladus, a high resolution mass spectrometer like the one going to Europa. Kelly Miller is in charge of the development of that for Enceladus. So I know I'm out of time. I'm going to just spend 5 minutes going over what we would look for. So first and foremost, we would want to identify the pattern of amino acids that are present. So on the left-hand side, you see the abundances of several amino acids for a meteorite, which is where life is, where the amino acids were not formed by life, versus an E. coli bacterium on the right-hand side of that gray chart. And the amino acids that are measured there are listed in their abbreviated forms in the little legend there. So in the abiotic system, glycine is the easiest amino acid to form. It has the lowest free energy. And so when that is dominant, you have an abiotic system because glycine is the least useful amino acid and is the easiest to form. But if you go to a bacterium, you see that glycine is less abundant. And indeed, in an abiotic system, the abundances of the amino acids replicate very, very closely the free energies of formation of those species, assuming there are no kinetic inhibitions. Whereas in biology, the abundances are determined by the utility of those amino acids. So that's a very strong difference in the biological and the abiotic signature of life. You can also look for the handedness of amino acids, not with mass spectrometry, but another instrument I'll mention in a moment. Biological samples have a very strong preference for one or the other handedness of these amino acids. They're, with the exception of glycine, they're not symmetric, they're chiral, so they have a mirror symmetry like your hands. And so that again is a strong indication of biological versus non-biological. So patterns like this are what we would look for. And in the mission that we are working on, we would do a number of tests with mass spectrometry, and we'd also do gravity, by the way, to understand where the ocean is and more about the density of the core, the silicate core. And our partners in that are Ozzy, and Antonio Genoa in particular is leading that part of the effort. So I'm not going to go through this whole flow chart, but we can ask the question, first, is the plume sourced from the ocean? And we can answer that yes or no. We can ask whether all the major biological building blocks are available. I think we've already answered that yes with Cassini. We can address whether there are sources of chemical energy with the mass spectrometry. We can even address whether the ocean is old or young with some of the non-biological or non-organic compounds that we would look at. And so we can determine how habitable that environment is. But by looking at the patterns of amino acids and the patterns of fatty acids, we can also do these tests to determine if those molecules are biologically generated or not. There's another instrument that we would fly, which is capillary electrophoresis. It would allow us to detect amino acids independent of the mass spectrometry but it would also allow us to determine the chirality of these amino acids. So this is the simplest first step in looking for evidence of biological activity in the ocean of Enceladus. And the next step, which could either be done NASA or ESA or both, but long after I'm retired or dead, would be something that would land on the surface and would collect more material than you could collect by flying through the plume. This concept is called Enceladus Orbulander. It was led by Shannon McKenzie at APL. It would land on the surface, open up a big collector, collect enough material that you could then analyze cells, if there are cells in that plume material, and actually do a form of genomic sequencing. So that's the story for Enceladus. I did have some slides on Titan. I'm not going to talk about those because I'm out of time. But I want to close, and we can talk about Titan in the refreshments. But I do want to close by pointing out that all of these life detection experiments that are proposed, and our group is not the only one proposing this, have to be able to determine the presence of false positives. We cannot allow for having a situation where we claim that there's biology present, but there isn't. We have multiple tests that all of which have to be positive in order to conclude that biological activity is going on inside of the Enceladus ocean. And the amino acid tests, the pattern of amino acids and the chirality are only two of those tests that we would do. So any good life detection mission has to have multiple tests for biological activity. It was Carl Sagan in 1996 in his final book who warned that while scientists' minds are open when exploring new worlds, and they have to be, because if we knew what was there before we found it, we wouldn't have to go. Nonetheless, we do have a talent for deceiving ourselves, and humans do have a tendency to discover the thing that we are actually looking for. And since we're looking for life, we want to make sure that we don't invent the presence of life. As Carl put it, there are wonders enough out there without our inventing any. And so the next generation of probes going out to the outer solar system, to these habitable oceans, to look for life, have to adhere to this warning of the great planetary scientist, Carl Sagan, who was an inspiring scientific hero of mine, and I know of many other people. So thank you, and I'm happy to take questions.
Of this fascinating topic. So I will ask you for the question. I will give the go to the people that are on the floor, and I will ask people that are remotely to raise their hand if they have questions, so we can manage for both of the questions. So yeah.
Very nice talk. Fascinating. The audience for another expertise.
Great.
Many decades of work. Two silly questions. One, where is all this water coming from?
Yeah, okay. So, yes, so the outer solar system appears to be very rich in water ice. Water, of course, is relatively easy to condense in the outer parts of protoplanetary disks based on measurements and also models of protoplanetary disks. And oxygen is a fairly abundant, in fact, it's one of the more abundant, most abundant of the heavy elements beyond hydrogen and helium. So we expect water to have been present in the planetary formation region, at least beyond the orbit of Mars. Now, there are complications to that story, whether the elemental oxygen is really combined with hydrogen to make water, or whether it's mostly combined with carbon to make carbon monoxide and carbon dioxide. But one only has to look at all of the bodies in the outer solar system, and with very few exceptions, they all have some amount of ice, water ice on their surfaces. And these happen to have liquid water because they have energy sources in their interiors that can melt the water. So in the case of Europa, it's a very large object. So it had a lot of energy of accretion, the virialized energy of collapse when it formed. Because it has a lot of rock, it has a lot of radioactive elements that are decaying and providing additional heat. And it has some amount of tidal heating from the Laplace resonance that it has with Io and Ganymede. And all of that adds up to making it easy for Europa to have liquid water. Enceladus is much more marginal. It's small and no one predicted it should have liquid water. It's not in a strong orbital resonance. It's in a near resonance with the moon Dione. And so the fact that it has this ocean is actually something of a mystery at this point.
Second question is about the methane out of the grid.
Yes.
So you mentioned that it was a very good issue. I'm not sure I follow
Oh, yes, okay. So, right, so I referred to methane being out of equilibrium at 2 points in the talk. The first point was when I talked about the Earth, which has an oxygen-rich atmosphere. And so methane is destroyed quickly in our atmosphere, and so it must be produced rapidly, and is produced, of course, by biology, in the case of the Earth. And you know, Mars seems to have some amount of methane, and why that's there is mysterious. Maybe there's life under the surface. In the case of the calculation I showed by Chris Glein, there, when you write an equation like that, where you have CO2 and CH4, and you have H2, you have water and H2, for the given temperature and acidity conditions in the ocean, you can of course calculate what the equilibrium ratios of all those species should be as the equilibrium constant, which is, you know, it's undergraduate chemistry to calculate that. But then you actually compare those abundances that you see with what you would expect at those temperatures, what the equilibrium constant would give you. And if the abundances of the methane, the carbon dioxide, and the hydrogen are not what you get from the equilibrium calculation, then you are somewhere either above or below the 0 point on that y-axis on the affinity graph. And so that's what I meant in that case in terms of being out of chemical equilibrium. And so on the Earth, any system, I mean, there are some microorganisms that are methanogens that produce methane in just that way. The feedstock, the carbon dioxide and the hydrogen, has to continually be supplied. Otherwise, they will drive their local environment to the equilibrium value, and then there will be no more energy available, and they will die.
But the current evidence is inconclusive.
For that calculation, it's inconclusive because it's actually the value on the y-axis that Chris derived is a little bit high for terrestrial microbes. And that either means that there really are not any there, or there are, but they are limited by the availability of one of the nutrients, like hydrogen, or maybe even, you know, some other element that they need to process the carbon dioxide. So the analogy I tell people is, for students, is if you walk into a restaurant and there are stacks of pizza there, it's obvious no one is eating the pizza. That's one possibility. The other possibility is that they're just limited in the amount of utensils they have, so they can only eat it slowly, and there's more coming out in the kitchen. You know, whereas in a system that's not limited by nutrients, people can eat it as fast as they can, and there are hardly any extra pizzas available. No one seems to like that analogy. I retract it, but I use it everywhere. So anyway, I hope that was illuminating and not confusing.
Other questions?
And yes.
Jonathan, you showed the gassing of Enceladus.
Yeah, gassing, yeah.
Do we have also some analog image of Europa, the gassing?
We don't, actually. So a very important question is whether there are plumes on Europa as well. Now, if there are, they're going to be much smaller than that plume on Enceladus because the surface gravity is a couple orders of magnitude higher for Europa. And so the plumes don't get very high up, they would be small. And one of the mechanisms for making those dark stains along the double ridge that you saw in those two places is small plumes carrying stuff out. So I think many people are familiar with the Hubble Space Telescope observation of excess atomic oxygen in the ultraviolet that seems to be concentrated toward the south pole of Europa. There are two different interpretations of that. One is it is from plumes, and the other is that there just is an ambient atmosphere, and you're just the concentration is not really significant. It's just part of an atmosphere. And we'll find out for sure from JUICE and Europa Clipper.
And that's interesting because in the image where you show the halo
Yeah.
around the fracture, actually the halo is just on one fracture, right?
Let's
or well, it's in the lake.
let's see. Which one are we talking about here?
No, in the one of Europa.
Oh, Europa.
Yeah.
Oh, okay.
Yeah, because the halo. Because I was wondering if
This one?
yeah,
Okay.
because the halo is
Oh, you mean that bright thing on the left side of the platypus?
no the dark halo around the fractures, like in the north.
The dark
part.
spots.
Oh, the dark spots. Well, I've seen
Also
them
the
on
next
both-
one
I've
you
seen them on
showed.
both sides, actually.
Yeah, but they are on the both side, but on the other fractures they are not.
Well,
It-
that's
Other
true.
examples you showed. other
Here's that
yes.
one. Okay, so
yeah, this one.
so this is the one from Galileo.
Yeah.
Well, of course, they only put arrows on the upper part, but I mean, if you come up and look at this image, this one looks symmetric on the left, and the one on the right also seems to be symmetric. So I'm not sure if it's one or the other, but you're right that the jets on Enceladus, for example, are not vertical, they're tilted. And so you would get preferential deposition on one side or the other for the material that falls back. Most of it escapes. For Europa, if you have a fracture where you don't have a vertical pole, then yes, you would get it on one side or the other. I don't know if that's part of their argument for this being the result of little plumes. It may be. But yes, in this case, with the higher gravity, anything that is not completely perpendicular to the surface would produce an asymmetric deposit. Yes.
And also, Jonathan, the observation was that some fractures do not exhibit, that do not show
That's
the
right.
dark.
They don't, right, exactly. So that, so this is also true for Enceladus, that in the case of Enceladus, the really high resolution images, the couple of images at high resolution, you can actually see that some of the fractures have material coming out, the jets, and others don't. The others are dead. So, whether they have sealed up or whether some of them open and close during different parts of the orbital phase, because Europa's orbit is eccentric. And so you would have a very significant change in the tidal stress regime as a function of position in the orbit. Some of those other fractures, maybe they open and close, but yes, you're right. Only there seems to be something special about a couple of those double ridges.
And
Yeah.
what about the molten part, the platypus? Are there any
platypus,
other
yeah.
examples? Or?
There are actually, and so I didn't take there's a nice comparison image from the paper that we published last year, but there are actually two or three really good Galileo examples, and they all have this sort of bilobed appearance to them. Some of them, the bilobe has a narrower neck to it, which is a larger separation, but they all seem to be bilobed or bilobate. And that doesn't necessarily come out of the salt, the brine melt model necessarily. But Yeah, there are these others that do resemble that. All about the same size. And all in this very heavily fractured region. Yeah.
I understand that the radiation environment in these objects is very tough.
Europa, yeah.
Do you see this as a stopper, as a breaker of the passage from radioactive to radioactive, or an accelerator of opportunity?
Right, that's a very good question. So the radiation environment at Saturn is much smaller than it is at Jupiter. So this is not so much of an effect for Enceladus. For Europa, the radiation environment, while of course it destroys organic bonds right at the surface, also generates oxidized compounds from the water ice. And so one speculation is that those oxidized compounds can make their way down into the ocean through the fractures and provide a source of chemical energy. And in fact, there are a couple of papers that calculate the amount of chemical energy available, assuming a certain delivery rate of these oxidized compounds. So too much would make it difficult for a prebiotic system where you just have organic chemistry to function, because the CH bonds would be broken and you'd end up with just more CO2 or HCO. But for biology, those oxidized compounds, hydrogen peroxide, for example, could actually be a source of chemical energy. So if there's biology today in Europa, that could be important. For the prebiotic situation, it could be an impediment, but the same was true the same was true for the Earth, right? If life formed in ponds on the surface of the Earth, they would have been subject to ionizing radiation, ultraviolet radiation, right at the surface, unless there was some shield, and there was not an ozone shield early in the history of the Earth because there wasn't much molecular oxygen. So we sort of have the same dilemma for the origin of life on the Earth, that some of the sources of energy that would be good for biology could be an impediment for the prebiotic reactions that would lead to biology. So I have not answered your question, but I think it's the same, in a way, it's the same problem for the Earth as it would be for Europa, at least. The problem for Enceladus is that there is no external source of chemical energy. And so you would expect that system to just grind to a halt. I mean, eventually all of the silicates would be leached out, everything would be hydrated. Why would there still be a source of chemical energy in this tiny moon if there's no open part to the system that provides energy? That's the real, for me, that's the real problem for Enceladus. Yes?
Maybe something correlated. You mentioned at the beginning that the molecular diversity or complexity is somewhat too large sometimes to allow for formation of life,
right,
biotic
yes,
activity,
right.
which seems to be counterintuitive. One would think that increasing the complexity and the diversity would give more opportunities.
Right, so
maybe it's the passage from chemistry to biology that gives.
Yeah, so I think it's a difference between complexity and diversity. So diversity by itself gives you a chemical system where it's difficult to select out those molecules that are most useful in the building up of biopolymers and catalysts that would continue to propagate the system, right? Just plain old diversity. So you throw chemicals into a chemical reactor, a big cooking pot, and you get lots of things, but there's no directionality. There's no way to select for the most functional molecules. Complexity is different because complexity implies that there is some level of information or structure. And so if systems evolve toward a more complex or more information-rich state, and DNA or RNA are sort of the epitome of an information-rich molecule, then you are evolving along the path toward life. and how that actually happened in the case of the Earth. How you pull out, for example, the most useful sugars, the ribose or the deoxyribose. How you separate out the left versus right-handed enantiomers from the amino acids in order to get the very first, even most primitive proteins. No one has figured that out yet. I think part of the problem is that it's very hard to replicate systems that have large and complex spatial scales, which would be these chemical environments, either at the bottom of the ocean in a hydrothermal vent or in some kind of Darwin's famous warm pond, which some biologists still invoke. It's very hard to replicate that in a laboratory, and doing it in a computer, that doesn't really simulate directly what's going on in a chemical system. It's too complicated. So I would separate diversity from complexity in that. Complexity is good and diversity might not be good. Because if you have 200 different kinds of sugars.
But how do you select these deposits?
That's the question. Absolutely. I think before I came here for my first, before I arrived here in 1998 for our first sabbatical, I was invited to a conference on physical mechanisms for creating chiral systems, which was held in Serra-Manzoni.
Settlements.
Yeah,
Yes.
that's right. And it was a fascinating meeting, and it was a beautiful town, but the answer it didn't seem like anyone knew more at the end of the meeting than they did at the beginning of the meeting. So that was a long time ago. Yes?
It's more curiosity, since you seem so involved in these topics, if about enceladus plumes, there is some numerical model that is trying to explain what happens in the subsurface, and in some way can explain this eject.
Well, there are mechanical plume models. Yeah. Are you talking about a recent one or just models in general?
Yeah.
So there are models in general, and so the model. There are a few different kinds of models, but the ones that seem to explain the velocity of the material coming out and the size distribution really just involve having a warm liquid water region where you open up a crack due to tidal stresses, because there are tidal stresses from Saturn. And so then you get water rising up because now you have a vacuum, and that water then begins to evaporate due to the vacuum, and the resulting jet of material coming out also drives out liquid water and particles and gas as well. So the closest analogy really is, and this was not mine, the closest analogy is the system for dumping urine from the International Space Station, where you have a bladder of urine, that is a tank of urine, and then you open a valve and it sprays out into space because it's a warm liquid suddenly exposed to a vacuum. And that generally is the model that people use for the plume, for the individual jets. And then those individual jets, as this material rises, the gravity is so weak that most of it escapes, it merges to form this larger plume that you see in the images. Does that answer your question?
Yeah.
Okay, if it didn't, tell me, and then we can talk more.
There's a question from remote. So, Marco Castellani, if you're there and you want to ask directly.
Yes, I'm here. Okay, just curiosity. Is that moon system between? Sorry, I have read my video. If we can have evidence of water.
Maybe you can ask.
Can you write it? Because you were breaking up. I'm sorry.
Yeah, also write it. So sorry, Marco, your voice is a little bit broken, so I will read what you write it. So it was just a curiosity. There are any other moon in solar system where we can still hope to find evidence of liquid water on the surface or under it?
Oh, okay. Is that okay? So I can answer that question. So there are other moons. I mentioned Ganymede, which almost certainly has an ocean under the ice crust. Callisto is a possibility. Those are the only ones for which there is some evidence. And for Ganymede and Titan as well, gravity measurements, the change in the mass distribution as they go around in their eccentric orbits is part of, for Titan at least, what gives us evidence for the ocean. And in the case of Ganymede, actually, it's the presence of auroras that it has its own magnetic field. There's an aurora, it seems to move up and down in a way that would suggest an ocean. Those are the only ones for which there's direct evidence. There are other bodies that are large enough that they could have underground liquid oceans, but there's no evidence one way or another. And that would be Triton and Pluto, and then the very distant Kuiper Belt object Eris, and maybe the Uranian, some of the Uranian moons, because Ariel is close to and may have passed through a resonance. And then I think either Titania or Oberon are big enough that they might have liquid. But those are all based on models. Did that answer your question?
Yes.
So I think there is no other questions.
I have a
Yeah,
maybe
sure.
a silly one.
Yeah.
So the project was for a lander, of course. Apart from the technical difficulties, why not a rover? Because it's not a fully developed biosphere, so you cannot expect to land it exactly on the spot when you can buy signals.
For Europa?
Yeah.
Right. Yeah, well, okay, so, you know, first of all, for Enceladus, you don't have to land exactly in the right place. If you're just collecting, so I should say that not all of the ice particles escape into Saturn orbit, only the ones that are smaller than a few microns. There's evidence from the BIMS instrument, part of which was built here, and Angeletta was the PI. You can see coloration around the South Pole, which indicates there are deposits. So you just have to land on those, no problem. Yeah, Europa is much more difficult. So yes, the Europa lander, the whole idea of landing a fixed lander right in the place where you've got a deposit of organics within reach of your arm. You know, the nightmare is the thing lands and for those online, I'm now pointing out of the room. The organic deposits are, 20 meters away or 30 meters away. Why not, right? So I just think the Europa lander, first of all, we'll know from Clipper if there are places where there's definitely deposits of organics. And then the question is, are they widespread enough in those areas that you could actually navigate and land and sample those? But wow, you know, rover on Europa with radiation environment, because the last study for the Europa lander had it functioning for, I think, 2 weeks before the radiation would destroy it. So the rover, and then, you know, the trafficability of a rover on an icy surface, I guess you would have to have crampons essentially on the wheels.
If you are landing on top of your spot, you are ruining it.
Right, yeah, but I think, yeah, I suspend judgment until we get some data from Europa. Clipper and JUICE, which will be 2030, 2031, and a few years after that.
Great.
Okay, thank you.
Thank you very much.